Hello, Mr. Sanger, live from the storeroom here with a pre-lab video for you. This lab is called Factors Affecting Enzyme Function. So the objective of this lab, quite simple, you're going to measure the effects of different factors on the rate of an enzyme catalyzed reaction. In other words, how do various factors affect the speed at which a reaction takes place? And the factors you're going to be looking at is the presence of an isomer, substrate concentration, enzyme concentration, and changes in pH. So here's some background on the reaction that we're going to be focusing on for this lab. So the enzyme involved is called polyphenol oxidase. Note that it ends in ase, as many enzymes do. And you're actually familiar with this reaction because this is the reaction that causes browning in fruit. So if you cut an apple and just leave it sit on the counter, it turns brown over time. Well, that is because polyphenol oxidase is at work doing this. What is that enzyme actually doing? Well, the reaction it's carrying out or catalyzing is it takes a substance called catechol, which looks like this, and it converts it into a different substance called quinone. Now, what's interesting about this is that catechol is clear while quinone is brown. So that's what causes the color change, is that we're producing this brown product. Now, the way we're going to measure the reaction rate actually taps into this color change that we're talking about. So we are going to use some fancy tools called LabQuest and colorimeters. And I happen to have a LabQuest right here in front of me. Can you see that in the camera? Okay, well, I don't know. This is why you need a cameraman, I guess. So you got this LabQuest, and it displays all the information that you're gonna need to work with, and then attached to that is this thing called a colorimeter. So the way it works is inside this colorimeter, light is going to be passed through the sample. And so here you have a little solution of something here, and light is being passed through it. And then the amount of light that is absorbed as it passes through the sample is measured. So we're measuring this thing called absorbance. So if you have a really, really light sample, lots of light will pass through it. If you have a really, really dark sample, much of that light is going to be absorbed and it won't make it through. So take a moment and reflect. If the sample is very dark, will absorbance be high or low? And hopefully you said high. A dark substance will absorb a lot of light, so not as much light will go through. So basically, that's what this device is doing. It's measuring how much light is able to pass through the sample. Now, if this reaction is occurring, if we're taking catechol and turning it into quinone, what should happen to absorbance? We're going from clear to brown. Take a moment. Should absorbance increase or decrease? And hopefully you said it should increase because, again, we're creating this brown colored solution, and so more light is going to be absorbed by the brown substance than was absorbed in the clear substance. So if all goes as we think, we should see absorbance increasing over time while this enzyme does its work. Okay? So to dive a little bit deeper on the colorimeter, you're going to be loading a sample. There's a little lid that pops up here, and you're going to be putting a sample in there using something called a cuvette. Now, this cuvette, I want to point out, you probably can't see it in the camera, but there's, if you feel it, there's a ridged edge. You can hear that little scratchy sound, and then there's a smooth edge. You're only supposed to touch the cuvettes on the ridged edge. Okay, so you're only touching the ridged surfaces. And the reason why is because light is going to pass through the clear substance, and if you touch it, your fingerprints are going to get all gunky and disrupt the absorbance of light. So you only handle it by the ridged substance. And then the other thing is you need to make sure to use a cover. So these little plastic covers, and you cover it up, and that's important because if you don't use a cover, it can spill in the colorimeter and then ruin it, and these are kind of expensive, so we don't want you to do that. So hold it by the ridged edges and use a cover. So here's what's going to happen on lab day. You are first of all going to set up the LabQuest for data collection. So your teacher is going to walk you through the steps of how to do it, but basically we're going to tell the LabQuest, this machine right here, that we want it to collect one sample of data every second for 120 seconds. Okay, so we're going to collect data for 2 minutes, which means if you mess it up, you've only lost 2 minutes of time, which is kind of nice. Then we need to calibrate the colorimeter. And again, your teacher is going to walk you through this step and how to do it. But basically, we need to tell it. We're going to let light pass through, and we're going to say, okay, this is a perfectly clear solution. We're going to define this as 0% absorbance. And in this case, our perfectly clear is going to be the control solution. Before any enzyme has been added, we want that to be 0% absorbance. We're defining that, and then we're also going to essentially turn off the light and define that as 100% absorbance, where it's just all black. So we're defining 0 and 100 so that we can kind of scale our data between those two. Once that's done, your teacher will turn you loose, and you're going to be on your own to begin to prepare your cuvettes. So you're going to get 5 or 6 of these little guys, and you're going to begin to load them up and get things ready. So there's specific instructions that tell you exactly how much to add, but basically your first cuvette is simply going to be a control group. So you're going to add catechol, and then when you're ready for data, the enzyme. You don't add the enzyme yet. Your second cuvette is going to be an isomer, and maybe you don't remember what an isomer is. An isomer, remember, is a molecule that has the same chemical formula, or 2 molecules that have the same chemical formulas, but the structure is different. And so here is our picture of catechol, our substrate. Here are 2 isomers, hydroquinone, and you can see that the OH groups are just sticking out in different positions than in the original catechol. And then the resorcinol is another one where the OH groups are sticking out. So we're going to give you one of those 2. Hydroquinone is the one we're going to give you. So it's just a slightly different shaped molecule, and we're going to see how that affects it. Then you're going to combine these 2. You're going to combine catechol and the isomer together and see what that does to reaction rate. You're then going to do one with altered substrate concentrations. We're going to add a little bit less catechol and see what happens. And then we're going to change the enzyme concentration, add a little bit more enzyme than typical, and see how that affects the rate. Finally, we're going to have you guys do a pH treatment where you use water of a specific pH. So if you perform this reaction in a pH of 2.5, how does that affect the rate? If you do a pH of 9, how does that affect the rate? So you're going to prepare all these cuvettes, and then when you're ready to go, you're gonna begin to collect data. So a couple tips about this. Again, don't add enzyme until the very last second. Your cuvettes, they're gonna have liquid in them. Only touch them by the ridge surface. And then you're gonna use these little tissue things called Kim wipes to kind of rub the sides down and get rid of any thumbprints that might be on there. And then finally, you're gonna cap the cuvette, put that cover on, and then you're gonna invert your solution to kind of mix it up, okay? So your cuvettes are ready to go. They're clean. You're only handling them by the ridge surfaces. When you're ready to go, you're going to quickly add the enzyme, drop of enzyme, bloop. You're going to place it quickly inside the colorimeter. And note, I don't know if you can see in this picture, but there's an arrow at the top, and you want to make sure that the clear side of the cuvette is in line with that arrow, because that's the way the light is passing through. If you put the ridge side in that arrow, then it's going to block the light and not be able to go, okay? So we add enzyme, we cap it, we invert, we put it in, we close it, and we hit play, boom, on this device, okay? And we have to be really speedy, because as soon as we add enzyme, the reaction is happening. So if we add enzyme and then sit around figuring things out, the reaction is going to happen and we're going to miss it. We're not going to actually get that data. So again, to recap, like really smooth procedure. You might want to practice a few times, but add drop of enzyme, bloop, cap it, invert, place in, close, hit play, and you're rolling. Okay, the reaction will happen, and theoretically, absorbance should go up. Okay, it's just a question of how much it goes up and the rate at which it goes up. So once you're finished with all of this data, you are going to transfer that data to a Google Sheet, and you're going to generate 3 graphs. And basically, you're graphing these so that you can combine them and measure the slope and see how the slopes compare. So the first graph will have your control group, your isomer treatment, and then the combined catechol plus isomer treatment. Your second is going to be a control group plus the reduced substrate concentration and the added enzyme concentration. And your third is going to be control versus the different pH treatments. So the task for today then is to complete a pre-lab exercise where you try and predict what is going to happen with all of these different treatments that you're doing. And a useful analogy when thinking through your pre-lab is to think of a fictitious enzyme called stick ACE. And what stick ACE does is it takes a toothpick and it snaps it in half. Okay, so imagine for your control group that you have a big pile of like hundreds of toothpicks sitting in front of you, and you sat for 2 minutes blindfolded, because enzymes obviously can't see or hunt, and you just kind of randomly grab a toothpick, snap, grab a toothpick, snap, grab a toothpick, snap. How many toothpicks were you able to break in 2 minutes? Okay, that would be your rate of the enzyme catalyzed reaction. And now we're going to carry out our different treatments. So for the isomer treatment, we're going to add some plastic toothpicks. Okay, so they're a little bit different shape, and they can't break as easily. And so now you're feeling around and you're breaking toothpick, but then all of a sudden you come upon this plastic one. Oh, that's not, oh, okay. Grab another one, break, and then, ooh, another plastic one. Shoot, doesn't break. So that's going to be like the isomer treatment. The increased enzyme concentration, decreased substrate concentration, I'll let you guys kind of talk through what that might look like in our analogy, and then same with the pH treatment. So kind of think through that analogy, and that might help you in making some predictions about what should happen in this lab. So that's it. Except it's not quite it. I did want to point out that I just want to re-emphasize these 6 science practices of AP biology and how we are certainly engaging in these. We are explaining the concepts and creating visual representations of how enzymes work and convert substrate to product. We're carrying out an experiment and asking questions about how different things will affect the rate. We're going to be representing and describing data by graphing this in Google Sheets. We're going to be analyzing that data, and then eventually we're going to make a claim about our results, and we're going to justify that with reasoning. So don't forget about those 6 science practices, and now I'm done.